Good morning, dear students. Today we are going to study about lactic acid fermentation. Lactic acid fermentation is also referred to as lactofermentation, and it is used to preserve dairy products, vegetables and meat for longer period of time before the invention of refrigerators or other food preservation techniques. It provides the final products with characteristics, aroma and textures, and it plays a crucial role in food safety and hygiene. Lactic acid fermentation technology is a cost-effective means of processing and preserving food with added values like better taste, better aroma and better texture. Microbes involved in fermentation. A lot of microorganisms are involved in the lactic acid fermentation, especially bacteria species, fungi and microalgae. And the bacteria involved are both heterofermentative bacteria and homofermentative bacteria. And this lactic acid bacteria species involved in fermentation are Lactobacillus species, Lactococcus, Streptococcus thermophilus, Leuconostoc, and fungi are Rhizopus and yeast and microalgae. And these microorganisms have the ability to convert sugars into lactic acid. And lactic acid produced by these microorganisms inhibit the subsequent growth of harmful bacteria, and it also provides favorable conditions for the yeast activity, which can be utilized for the commercial production of beer and wine. So while we are using microorganisms to produce wine and beer, we are applying, or we are using, especially we are using Saccharomyces cerevisiae for the production of this wine. So while we are using the yeast for the production of wine, we can apply a little quantity of lactic acid, so it will provide favorable conditions for the growth of this yeast, and it will help in the better production of this wine and beer. So this bacterial or this microbial fermentation, it accounts for about 90% of the total lactic acid production in the world. And next is the raw materials involved in the production of lactic acid. So many raw materials can be used for the production of lactic acid, and it include both pure substrate as well as waste products of industries, and also starchy materials. And if we are using the pure material, then the cost of purification is very less. And the pure substrate commonly used are sucrose from sugarcane and sugar beet, and also starchy materials such as wheat, corn, maize, cassava, potato, rice, rye, barley. They are all very potential raw material for the production of lactic acid. And also waste products from food industries, agricultural industries, sugarcane mills and biomasses can also be used as a raw material or substrate for the production of lactic acid. Industrial lactic acid fermentation. So microorganisms, they will act on these raw materials, and they have the ability to convert sugars present in the raw materials into lactic acid. So C6H12O6, that is the glucose, that is get converted to C3H6O3, that is lactate plus carbon dioxide. The production of lactic acid occurs through following step if lignocellulosic raw materials are used. These lignocellulosic raw materials are present in woody materials or woody parts like this sugarcane waste. The lignocellulose will be the raw material, and so the microorganism has to follow, or before this microbial oxidation takes place, we have to pre-treat the lignocellulosic material for the industrial lactic acid production. So the first step is pre-treatment of the lignocellulose to break the complex lignocellulosic structure. Second step is enzymatic hydrolysis to depolymerize lignocellulose to fermentative sugars, and third step is sugar fermentation by microorganisms to lactic acid. So these three steps has to be followed if we are using lignocellulosic raw materials like sugarcane, molasses, like etc. Next is lactic acid bacteria. So when we are using, when we are commercially producing this lactic acid, we are using mainly lactic acid bacteria or yeast as the microorganism. Both homofermentative lactic acid bacteria and heterofermentative lactic acid bacteria have the capacity to convert sugar to this lactic acid. But when we are using this homofermentative lactic acid bacteria, it will convert sugar, almost all the sugars, to lactic acid. But if we are using heterofermentative lactic acid bacteria, then the glucose is converted to lactic acid, but along with the lactic acid we will obtain ethanol and carbon dioxide. So the yield will be less when compared to that of homofermentative bacteria, because along with the lactic acid it will be producing a little quantity of ethanol and carbon dioxide. So the productivity of lactic acid will be less when compared to that of this homofermentative LAB. Then homofermentative lactic acid bacteria, that it will normally metabolize glucose into this lactate by glycolysis or embedded Meyerhof pathway. So homofermentative bacteria will directly depend upon this glycolysis for the production of lactic acid from glucose. Here two molecules of lactic acid are or two molecules of lactic acids are produced from a single molecule of glucose, which result in an yield of more than 0.90 gram per gram of glucose. So almost all the glucose, that is for 1 gram of glucose we are getting almost 0.9 gram of lactic acid. So a high proportionate conversion of glucose to lactose will be lactic acid will be taking place if we are using homofermentative lactic acid bacteria. And so many homofermentative lactic acid bacteria are available, and among them we are mostly depending upon Lactobacillus acidophilus, Lactobacillus bulgaricus and Lactobacillus lacti. These three bacteria commonly used homofermentative lactic acid bacteria: Lactobacillus acidophilus, Lactobacillus bulgaricus and Lactobacillus aceti. Lactobacillus aceti is not mentioned here, but Lactobacillus aceti is also being used for the lactic acid fermentation. So homofermentative lactic acid bacteria and heterofermentative lactic acid bacteria are there. But for the commercial preparation of lactic acid, homofermentative lactic acid bacteria are much better, because it will yield much proportionate of lactic acid when compared to that of heterofermentative lactic acid bacteria. Next, parameters that affect the fermentation process. So we have already completed the raw materials and microorganisms involved, and then we have to focus on different parameters that affect the lactic acid fermentation, both pH, temperature, then and nutrients like nitrogen and vitamin sources, and fermentation operation and byproduct formation all influences the lactic acid fermentation process. And the most suitable temperature will be ranging from 30 degree to 43 degrees Celsius, and the nitrogen sources and vitamin are very important for this bacteria to synthesize this lactic acid. So we have to provide nitrogen sources and vitamins along with the raw materials, because the bacteria, this lactic acid bacteria, they have a limited ability to synthesize the B complex vitamins. So we have to supply the B complex vitamins along with the nitrogen sources. So the main nitrogen sources are yeast extract and ammonium sulfate. So yeast extract and ammonium sulfate are the main nitrogen sources that we are applying along with the substrate, and we have to add vitamin B also for the fermentation to take. Next parameter is the pH. pH of the fermentation decreases according to the amount of lactic acid produced in the fermentation process. So as and when lactic acid get accumulated, the pH of the medium will get reduced, because lactic acid itself is an acid, but if the pH of the fermentation medium get reduced, then the lactic acid production will be 0 or minimum. So in order to enhance the production of lactic acid, a neutral or only a slightly acidic pH will be required. So bases should be added to increase the pH when the lactic acid production increases. So bases such as calcium carbonate, calcium hydroxide, sodium hydroxide, etc. has to be added into the acidic medium as lactic acid production increases in the medium. And a pH of 5.7 is optimum for lactobacillus strain, whereas which are known to tolerate lower pH than lactococci. So lactococci, it will require higher pH, but lactobacillus strain, they can tolerate lower pH and they can tolerate up to 5.7 pH. So the 5.7 pH is optimum pH that has to be maintained if we are using lactobacillus strain for this fermentation process. Next is the different types of fermentation types that can be used for the production of this lactic acid. Both batch fermentation and continuous fermentation can be used for the commercial production of lactic acid, and both the process have their own advantages and disadvantages. Fermentation in batch mode has superior conversion and yield compared to that of continuous fermentation, but the volumetric productivity is lower. In a batch process, all the substrate get used, whereas in continuous process there is a residual substrate concentration that is always present. Highest productivity possible for continuous fermentation is due to the high dilution ratio and the possibility of maintaining the process for a long time. So if we are using the batch fermentation, then there will be a superior conversion and yield. That means almost all the substrate will get converted to the product, that is lactic acid. So there is no residual material in the fermenter. Almost all the sugars or the starchy material that we are giving as the raw material get converted to the lactic acid if we are using a batch type fermentation process. But the yield will be lower when compared to that of continuous fermentation. In case of continuous fermentation, the yield is higher, but some residual substrate will be there. All the raw materials will not be get converted to the product, and the reason is because this continuous fermentation is a continuous process and it will take longer period of time. So yield will be maximum for the continuous fermentation process. Next is fed batch fermentation. Fed batch is actually a type of batch fermentation process itself, but it has some slight modification when compared to this batch fermentation. This fed batch fermentation is most commonly used for the large scale production of this lactic acid when compared to that of batch fermentation and continuous fermentation. In fed batch fermentation, all the required raw materials such as carbon source, nitrogen source and other vitamins are added during the fermentation process at regular intervals of time without removal of the fermentation broth. So in fed batch fermentation, we are initially supplying the raw materials, carbon source and nitrogen source and other vitamins and other requirements into the fermenter, and these components are added at regular intervals of time without the removal of the fermentation broth. This type of fermentation is especially useful to maintain a low substrate concentration by supplying nutrient to the fermentation culture in regular intervals, which in turn reduce the substrate inhibition. So this is very essential in maintaining a low substrate concentration at the initial time. At the initial phase, we are not adding all the nutrients and the substrate. We are adding a fixed amount of nutrients and other raw materials at the initial phase, and at regular intervals we are enhancing the substrate as well as nutrient concentration. So the substrate inhibition can be avoided by this fed batch fermentation. Lactic acid production by lactobacillus using fed batch fermentation was found to be more efficient in production of lactic acid than other methods. So if we are employing this lactobacillus bacteria for this lactic acid production, then fed batch fermentation is the best method. And lactobacillus lactis for the production of L-lactic acid produced about 97% of yield of lactic acid at a rate of 2.2 gram per liter per hour. So if we are using lactobacillus lactis in particular as the bacteria for the production of L-lactic acid, then we can obtain 97% yield of L-lactic acid at a rate of 2.2 gram per liter per hour. So fed batch fermentation is the best method for the commercial production of lactic acid using lactobacillus lactis as the bacteria. Next is separation and purification. So after this lactic acid has been produced, then we have to separate the lactic acid from the fermenter and we have to purify it. So a lot of downstream treatments such as precipitation, conventional filtration, acidification, carbon adsorption, evaporation, crystallization and others takes place in order for the separation and purification of the lactic acid. And these all processing steps will influence the quality as well as price of the product. Uses of lactic acid. Why we are producing this lactic acid in a commercial scale. Lactic acid, it is a naturally occurring organic acid that can be used in a wide variety of industries such as cosmetics, pharmaceuticals, chemical, food and medical industry. So lactic acid, it is having a wide variety of application in so many industries like cosmetics, then pharmaceuticals, chemicals, food and medical. And it is used as an aciduant because of its mild acid taste when compared with other acids used in food. So actually what is lactic acid? Lactic acid is produced from lactose. So milk is a good source of lactose, and when bacteria, when especially lactobacillus, when grow in milk, it will convert the lactose to lactic acid. So lactic acid is actually the curd. So this lactic acid, it is having a mild acidic taste, and so it is used very commonly in food to preserve. It is very commonly used in food as a preservative, especially for olives and pickled vegetables. And it is used as a flavoring agent, a pH regulator, an inhibitor of residual bacteria in food processing such as in sweets, bread, soft drinks, beer and other products. So this lactic acid, it is used as a preservative agent for pickles. It is used as a flavoring agent. It is used as a pH regulator. It is used to inhibit growth of other bacteria in food like sweets, bread, soft drinks, beer and other products. And it is also used as an essential ingredient in the fermented food like yogurt, butter and canned vegetables. And it is also commonly used for the production of polylactic acid or PLA in industry. Polylactic acid is a biodegradable plastic that is been prepared from this lactic acid, and it is it has the same purpose as that of the normally used plastic. So the wide variety of uses are there for this lactic acid. So that is for today class. Thank you.